Welcome back. I'm Dr. Elise Martin. By now we've explored digestion, peptides, methylation, neurotransmitters, circadian rhythm, adaptive monitoring, and entrainment. We've seen how autism is shaped not by a single failure, but by interacting systems that slowly fall out of rhythm. In this chapter, we bring several of those strands together. The digestive tract is often described as plumbing, but biologically it behaves more like soil. It supports communities. It responds to pH. It selects which organisms thrive and which decline. Microbes do not randomly populate the gut. They are shaped by acidity, bile flow, oxygen gradients, motility, and nutrient availability. Change the environment, and the community changes with it. This is why lasting microbiome repair cannot be achieved by adding organisms alone. The habitat must support them. pH is one of the most powerful regulators of microbial balance. In the stomach, acidity activates pepsin and initiates protein digestion. In the small intestine, pH shifts trigger secretin, bile release, and pancreatic enzymes. In the colon, slightly acidic conditions favor beneficial short-chain fatty acid-producing organisms. When pH regulation falters, microbial selection changes. Opportunistic species gain advantage. Beneficial fermenters decline. Metabolite profiles shift. This is not infection, it is ecological drift. Many gut microbes participate directly in neurotransmitter metabolism. They influence tryptophan availability, serotonin synthesis in the gut, GABA production, and dopamine precursor balance. When microbial populations shift, so does neurotransmitter signaling, not only locally, but system-wide. Importantly, most serotonin is produced in the gut, not the brain. When gut ecology is disturbed, serotonin signaling becomes erratic. This contributes to mood instability, sleep disruption, altered pain sensitivity, and changes in motility. Once again, behavior reflects ecology. Acid suppression is often used to manage gastrointestinal discomfort, but when acidity is chronically reduced, several downstream effects follow. Protein digestion becomes incomplete. Peptide load increases. Secretin signaling weakens. Bile flow declines. Microbial balance shifts upward into the small intestine. And over time, this alters the entire microbiome pH neurotransmitter axis. The system adapts, but not optimally. This adaptation often includes increased somatostatin tone and reduced signaling amplitude. Microbes influence motility. Motility influences microbes. When gut movement slows, bacteria have more time to ferment carbohydrates in inappropriate locations. Gas production increases. Inflammatory signaling rises. That inflammation further slows motility. This loop sustains dysbiosis even when diet and supplementation improve. Breaking the loop requires restoring rhythm, not just killing organisms. Neurotransmitters do not act only in the brain. Serotonin regulates gut motility. Dopamine influences immune tone. GABA modulates visceral sensitivity. When neurotransmitter signaling is unstable, gut function destabilizes. And when gut ecology is unstable, neurotransmitter production fluctuates. This bidirectional loop is why gut symptoms and behavioral symptoms often rise and fall together. They are not separate problems. They are different expressions of the same axis. Probiotics can be useful, but they are not ecological architects. They introduce organisms into an environment that may or may not support them. If pH, bile flow, motility, and nutrient availability are unfavorable, introduced organisms simply pass through or fail to colonize meaningfully. In some cases, they even increase competition and instability. This is why probiotics work best when the underlying environment has already begun to normalize. They reinforce balance. They do not create it. Stabilizing the microbiome pH neurotransmitter axis requires order. First, restore acid and digestive signaling so proteins and nutrients are processed fully. Second, reduce peptide and inflammatory burden so immune signaling quiets. Third, support bile flow and motility so clearance resumes. Fourth, allow microbial populations to reorganize naturally. Finally, neurotransmitter balance has an opportunity to stabilize as a result. This sequence does not rush the system. It allows it to reselect equilibrium. When pH rhythm is restored, microbial ecology stabilizes. When microbial ecology stabilizes, neurotransmitter signaling becomes predictable. When neurotransmitter signaling becomes predictable, behavior stabilizes. This is not symptom control, it is ecosystem repair. And ecosystem repair tends to hold, because it reduces the need for constant compensation. This content and other content on this channel is for informational purposes only, and should not be considered medical advice, diagnosis, or treatment. Always consult with a healthcare professional before making any changes to your health routines or treatment regimen. I would like to thank you for sticking around through the full episode, and hopefully you found some value. Please share, like, and subscribe for the benefit of everyone in the ASD community. As we close this chapter, I want to leave you with a broader perspective. The gut is not simply where food is digested, it is where chemistry, ecology, and signaling meet. When we restore the internal ecosystem, respecting pH, rhythm, and microbial balance, the nervous system no longer has to work so hard to regulate itself. In autism, stability often comes not from adding more inputs, but from restoring the environment that allows balance to emerge naturally. In the next chapter, we'll explore how synergistic stacking and timing can support this ecosystem without overwhelming it, and how carefully sequenced interventions can amplify one another rather than compete.